So virus is unlike bacteria, you all know, it's very different. And for example, bacteriophages, they will grow on the agar, but animal viruses, they don't grow on the artificial what you call material like agars, but they really need a host, living organism. That's why it's very difficult to work with virology viruses. So you need always a living host system so that it will grow on it. And it mostly doesn't require antibiotics also, unlike we use streptomycin in our regular bacterial, but yeah, here it doesn't require. And there are mainly three methods for cultivation and culturing of viruses. One is laboratory animals. The other one is embryo-oriented egg. I mean, it's not a, it should be embryo-oriented egg. And the final one is cell culture, like living cell lines, like zero cell line and hectonate cell line, etc. And after culturing, we have different methods to detect whether virus is growing or virus is present. That is measured by one of the method is cytopathic effect. We can use the immunofluorescence method to measure how it is growing. I will explain one by one in coming slide. Yeah. So the simple one, what we learn in our department is cultivation is the egg. It should be embryo-oriented egg. And this method was developed by Posher and Burnett in 1913, and they used this simple what we call a model for growing the viruses. The egg should be very sterile, and the shell should be very intact and healthy. If there is some damage, it will interfere with the virus culture. And they're using embryo-oriented chicken eggs, duck, and also turkey eggs for this kind of culturing, especially they use for influenza virus purpose. And the egg should be 7 to 10 days old embryo-oriented egg. As I said, it should be cleaned before going to culture with a disinfectant, and the shell should be decontaminated with the as I said, disinfectant, and checked with the ovoscope. Ovoscope is a method to check that the embryo is alive, so that virus can grow. We call it candling also. Yeah. So how cultivation is happening in embryo-oriented egg? There are different routes where you can inject the virus, but this is also depending on the type of virus. There is one I think I made that slide here. Yes. So you can see here right side, the egg has different parts inside. For example, amniotic inoculation, yolk sac inoculation, and allantoic inoculation. So each different types of virus will prefer the different location of the egg. That's why scientists have developed, for example, influenza virus and mumps virus, they only grow in a amniotic fluid and amniotic location. And herpes simplex virus is growing in a yolk sac. Like that, they have standardized and optimized. So location of the egg is depending on the virus. So yeah, this is the methods we use. And if you take, this is also you can find in Google, the embryo-oriented egg has a different parts, and this is a needle with the virus inoculation. They may basically drill it, and also you can do the manual puncture. We was doing manual puncture before, and then you, depending on the location, you are inoculating the virus. And see here, by using this, we call it candling. Different days you can measure whether the egg is the embryo, how the embryo is developing, and based on that we can also see our virus is growing. So this is the practice, old practice, people were used to do it, and now I think this were maybe small amount of viruses, and for demonstrating the lab is fine. But if you want to culture the virus in large amounts and for the experiments, I think people are going with the animals, and also the like tissue culture. So the next question is, how do you detect the virus? Well, after inoculating into the egg, what are the signs? How do you measure that? I think the signs of viral growth include, like, once the virus is growing very intensely, means that you'll find that the embryo will be dead. The death of embryo is one of the signs. And defects in embryonic development, that by, like I showed before, we are measuring the embryonic development here. So if the embryo is not developing properly, means the virus is growing nicely. And You can also measure by damage in the localized areas like membranes, and also different signs like resulting in a discrete and opaque spots. We can see the spots also called pox. Yeah, by all these signs we can see that the virus is growing in the embryo. And then the embryonic fluid and tissue can be prepared for examination of the electron microscope. So if you further want to see a virus levels, I mean virus in the egg, you can just take the sample and you can go for the electron microscope. Yeah, and there are other methods like you can we can see by agglutination of red cells, etc. I'll explain later. So the next one is, as I said, growing this. For example, let's take SARS, I mean COVID-19. How people are growing. Recent videos I can show on the animal cells.